Okay, well, first of all, thanks very much for inviting me. I'm really excited to learn from all of you here today. I think the range of perspectives on the trait approach to understanding microbial diversity represented in this room is pretty amazing. And yeah, as I said, I'm very excited to be here. I'm going to talk a little bit about the genomic origin of microbial traits, primarily in soil and subsurface systems, but the concept should apply to any type of ecosystem. But one of the reasons I like to talk about soils is their complexity. And we're dealing with a situation where we have incredibly complicated biology, maybe you could call this complex as well, interacting within complex systems, and complex systems being systems that are open, interacting, and full of non-linearities. And the challenge here, I would say, unlike approaches to trait-based modeling in vegetation, is one, we typically can't see what we're working on. It's very hard to measure the traits, and the extent of metabolic diversity is enormous. We're not just dealing with phototrophic organisms, we're not just dealing with one or maybe mixotrophs. We're dealing with organisms that can use a wide range of electron donors, a wide range of electron acceptors, and then the necessary physiologies that goes along with those types of metabolisms. So this phylogenetic diversity of the microbial, or actually the tree of life, also extends to amazing metabolic diversity. So how do we deal with this? Well, we've just heard we have the metabolism of the inner workings of a cell. We've got cells interacting here. I just heard at lunchtime today that in the human microbiome, unlike in soils, interactions can be very intimate but also very infrequent. Cells can be separated by quite large distances. The structure, the porous matrix at which organisms interact is extremely complicated, and that results in a lot of coexistence of different metabolisms because you have spatial separation, and then gradients, thermodynamic gradients, can form at this scale. But we also have drivers at larger scales, such as this is an aggregate here, and at the rhizosphere, a growing plant root introduces resources, and this resource pulse stimulates organisms to cross-feed, to compete. So you've got both competitive, mutualistic interactions going on, antagonistic. And then at the larger scale, we have these drivers, hydrology, topography, climate, etc., that regulate these interactions, but also regulate interactions down to metabolism. So extremely complicated set of scales to be able to span. If we want to be able to understand the consequences of traits at this scale to processes at this scale. Okay. So the plant community has sort of come to a similar conclusion, the vegetation community, of moving from plant functional types to trait-based models because of the diversity of features of vegetation that are not captured in plant functional types, and also, as we heard earlier on today, the need to build models that are emergent, that allow us to identify when and where specific combinations of traits are valuable to an organism and improve its fitness. So moving from microbial functional types, which is the typical approach in the types of models we use, reactive transport models, moving from that to a trait-based approach for the same reasons. Okay. And I think you've probably, I'm preaching to the choir here at this point. There's a range of different approaches here, from individual-based models up to stoichiometric metabolic models. We sort of work in this space in the middle here, where we're abstracting some features of metabolism, but not at the whole community, still representing trait distributions at the species or potentially functional guild scale. Our approach here is to quantify trait distributions, the linkage of traits, and to use a thermodynamically based performance filter. So these are reactive transport models, so that we can allow environmental gradients to form at the scale of the microbe. So I think you've probably all seen a version of this in Mick Follow's papers. The physiology as predicted by either experiments or from the genome, interacting with the environment, regulating fitness, impacting community assemblage, which in turn the emergent property of this is a reaction rate contributed to by many organisms, which in turn feeds back in the environment and alters the fitness. Okay. So what sort of traits do we mean? Again, this is probably preaching to the choir here, but when I present this to non-microbiologists, I start with an analogy. Here's an automobile. So we might want to know something about the engine type, in our case, our metabolism here. In some cases, we don't need to know all of the details of the engine type. We just need to know it's a certain type of engine. We need to know something about the intake, the uptake, the size and the shape, the materials it's made from, the fuel type, fuel tank capacity, exhaust system, things like that. And with that, then we would predict things like weight, power, top speed, maximum range, efficiency, and emissions. So to extend that analogy to microbes, well, maybe we're interested in energy yielding pathways, whether it's respiration, fermentation, photosynthesis, maximum specific growth rate. Are cells wired for speed? Is that part of their evolutionary history? The kinetics of uptake, substrate uptake, etc. Nutrient use efficiency, whether they make storage compounds, what their preference for donors, acceptors, etc. All the things, size and shape, allometric properties, biomass stoichiometry, which is emergent, optimum temperature, pH, and production of things like extracellular enzymes. And with that, again, we'd like to represent growth rate, biogeochemical reaction rates, emissions, and then the feedback of those individuals to the environment. So the next I want to step through some examples of how we go about quantifying these traits, and an approach that I think, something that I think about at least anyway. This is our challenge. We can't cultivate most organisms. We will never cultivate most organisms. So we have to work together as a community to build generalizable understanding of the distribution of traits and validate the accuracy of those predictions. So for the time being, we're limited to various omics methods, whether single cell, metagenomes, transcriptomes, proteomes, metabolomes. These lots of things that are increasingly less expensive to do give us some prediction of traits and the trait linkage, but really these are hypotheses. That's all they are, about the roles of genes, the roles of organisms, and depending on the location they came from, the conditions they came from, some environmental context to relate the traits to. So trait distributions with some estimate of maybe what environmental filters may be important influencing those distributions. The other thing we need is realistic measurements of the environments that the organisms are in to interpret those trait distributions. This brings us to approaches such as imaging and sensing, not just of the biology, but the physical environment and the chemical environment in which the microbes are competing, cooperating, interacting. And again, at the right scale. This then gives us information on the environmental filters of the traits, what sort of gradients do organisms align themselves across, and what sort of co-associations are more common. And again, that gives us a sort of a better picture of the fitness of an organism, but not in isolation, in cooperation or collaboration with organisms that it consistently finds itself with in the environment. And then really, again, we're building a body of evidence along here, and what we want to be able to do then is to demonstrate the importance of these traits, and so manipulation here is key. So performing experiments or working in the real world, creating realistic physical and chemical environments, such as constructed ecosystems that mimic the key properties of the environment that you're trying to replicate, for example, in soil, advective versus diffusive domains, minerals with surface reactivity that affect the availability of nutrients, etc., and affect the interaction space between organisms. Also cultivation, and I can't stress this more strongly, our ability to ground truth, these hypotheses that we're making from omic approaches. Cultivation is key. It'll never get us all of the way, but it'll get us at least at some verifiable linkages between genomes and traits, and realized traits. So this, again, with cultivation then, a range of genetic and chemical tools become available to create mutant libraries to identify the specific genes involved in fitness under given conditions. And all of these things are becoming higher and higher throughput, so that I think this is looking promising. So these are technologies that give us, I would say, knowledge, or the beginnings of knowledge, and then enabling prediction, such as generalizable patterns of microbial metabolic potential, what traits are consistently linked, defining sort of reaction networks for systems, what are the major reactions in a system that dictate the interactions between organisms, and then with their manipulation, really validating the roles and feedbacks that individual organisms, individual nodes in an interaction network, how they impact the system. Okay. So I want to give one example first. This is a merger of cultivation and omics in the rhizosphere, and this is work by Kate Zelnina, who's a postdoc who works between all of us here, but primarily Trent Northern and myself. And this is work in a Mediterranean grassland where we're looking at the rhizosphere microbiome growing with an annual grass. So what we observed before, previous work looked at rhizosphere microbial community succession by sampling exactly right by the root, so defining what we think is the rhizosphere, over 2 different seasons, just looking at the patterns of microbial community succession. And what we noticed, this was Xingjing Shi published this, is that here's season 1, here's season 2. These are bulk soils that were not influenced by the root, and this is the succession of the root microbiome. What you can see here is that on this axis here, they follow the same pattern. So reproducible. They're different across this axis here because these ones have the dead roots from the previous year. So there's some difference here, but the successional patterns are the same according to rhizosphere development. What we also noticed, these are different types of organisms. You probably can't see this here, but the dominants in the system are actinobacteria and alpha proteobacteria. Actinobacteria consistently went down, at least by this measure. Alpha proteobacteria consistently went up. So we've got positive and negative responders for the rhizosphere. We want to know why. Okay, so, well, there are a number of possibilities here. This is consistent. Is it related to things like growth strategy? The dogma is that copiotrophic organisms might grow more rapidly in response to a resource pulse in the rhizosphere. Okay, so that was the first thing to look at. Maybe they've got different organisms have different metabolic requirements, different substrate preferences, exudates versus polymers. That's talked about quite a lot. Other traits, motility. Can they swim to the rhizosphere? Do they need to signal to the plant to be able to reside in the rhizosphere? I'm not going to talk about this today, but we've heard a great talk by Ali looking at interspecies interactions, antagonism, cross-feeding, facilitation, etc. These are all likely important. Before I go on, any bets on what's important? Growth strategies. Resource utilization. Everything? All? Okay. All right. Okay. So we've one of each. All right. Let's see. Okay. So we isolated Ulysses when he was in our group, isolated a whole load of bacteria, confirmed that they were numerically relevant and similar to bacteria that we had identified based on 16S libraries. So they're relevant OTUs at least. We sequenced 39 of those genomes. And then we're able to map those organisms onto OTUs that responded positively or negatively. So these are colored here based on green are bacteria that responded positively to the roots. These magenta ones are ones that responded negatively to the roots, and this is statistically across replicated experiments. One thing you'll see is there's a bit of a phylogenetic signal here, but that doesn't explain all of the variability, which is good. Okay, so these isolates allow us then to test genomic hypotheses that are related to physiological traits. One thing that we're very keen on is maximum growth rate, or predicting new max using codon usage bias. Ribosomal RNA copy number is another good predictor for that. We found this seems to be best in our case, but also optimum growth temperature based on purine pyrimidine dimers. And when we look at this from our isolates, we can see that we span quite a range of different minimum generation times, quite a range of temperature and growth optima, and there's also a bit of a phylogenetic signal in that too. So let's go back to this. So now we're looking at genomic traits in the rhizosphere. One of those is generation time, the positive versus negative responders. Positive responders are slower growers. Based on their life history, they're slower growers, not faster growers. So the copiotroph idea seems like it's not. Remember, this is just a genomic prediction though, so it doesn't seem to explain it. They've also got bigger genomes. The positive responders to plant roots invest less of their genome in glycoside hydrolases for depolymerizing. So the ones that live in the bulk soil seem to do more of that, or have the potential to do more of that. But when we look at transporters, the positive responders for total transporters, amino acid, organic acid transporters invest a significantly greater real estate in their genome for uptake of these monomers. So the higher numbers of transporters in genomes, particularly amino acid and organic acid transporters, lower number of glycoside hydrolases in the positive responders, but wired for slower growth. Okay, we tested this in the lab because we had the isolates, and this is measured versus predicted for growth, maximum specific growth rate. There's some anomalies, but it does a pretty good job. And as you can see, The red and the green here are the positive and negative responders. That does not explain the response to root growth. Being able to grow fast does not explain the response to root growth. So is the succession in, go ahead. Yes, these are all cultivated, grown under a range of conditions in the lab. So we may not have hit the maximum growth rate. This is the growth rate maximum that we could obtain in the lab, but we may not have hit their maximum growth rate. That's always hard to prove. But we got close to the predicted, so we stopped there. Is succession related to resource dynamics? Well, one thing you can do then is look at the resource dynamics in the rhizosphere. Here is total organic carbon. This is Avena grown hydroponically. So here we're looking at just programmed processes in plants. So there is a nutrient limitation similar to what we would expect in the field here in terms of lower nitrogen, but other than that, it's in hydroponics. And you look at we see an increase in total organic carbon released that peaks around about 9 weeks during plant growth. Okay, then you can look at the composition of this, and this again is work in collaboration with Trent Northern's lab, doing metabolomics on the exudates. And so we can see here's an ordination of week 3, week 6, week 9, and week 12. And so the composition of the exudates changes over time as well as the quantity of the exudates, and particularly organic acids, amino acids, nucleotides, etc., peak at week 6 and week 9 when we have the higher concentrations. Okay, so all right. So does the rhizosphere resource dynamics drive microbial succession? In that case, that would connect the traits of increased transporters to the traits of the plant in terms of the chemistry that the plant is releasing. So remember, our transporters were up, our GHs were down. Okay. So Trent's lab has developed this approach called exometabolomics. We take the exudates from all different time points from the plant, pool them together, make a medium at them, feed them to the different microbes, and then see what's eaten. So things that are eaten and the amount that's eaten is we treat that as a preference, and there's a score for that preference. So that tells you whether organisms have a preference for organic acids versus etc. Okay. And when you look at this, so here what we're looking at here is percent uptake from the medium, and again positive against negative, and we see a significant shift in the distribution for amino acids, organic acids, and a less substantial but also significant shift in sugars. So that tells us that the organisms that respond positively to the roots have a greater potential for uptake of amino acids, organic acids, and sugars, which corroborates our prediction from the transporter, the allocation to transporters in those cells. So with that, we're now moving towards building a trait-based model based on Jin Yun Tang's model and working with Bill Riley that represents substrate preference, growth rate, temperature. I didn't show temperature data here, but temperature, optima, etc. Okay, so that's sort of the first story, but I want to throw a caveat in here while we're doing this. I've talked about OTUs and relating OTUs to patterns that we're observing in the rhizosphere. And so I want to say beware of OTUs. What we also have observed is niche optimization at the species or strain level. So while we have positive responders, and those positive responders, we think we have genome predictions for why they're positive responders, there are also other traits that are important that are, you might call subtleties, but are very important. Here are 3 organisms that are similar to each other. They're all Bradyrhizobia, probably different. They are different. Genetically, they're probably different species. But if you look at their 16S identity, they would all fall into one OTU, 97%, 99%, 99% similar. So we would bunch them into an OTU, and we would follow their dynamics together. And when you're looking at this here, each color is one of these different sequences. Some of these are isolates, and one of them is a metagenome, reconstructed genome. So some of them do go up in response to the plant, others stay the same, others go up and go down. And when we're doing 16S analysis, we're averaging all of those together. And if we're trying to infer traits of organisms while we're averaging those together, then our response of an OTU group is being averaged. We're potentially losing signal. So, okay, you guys probably you already get this, but I just wanted to make that point. Okay, they have different responses to root growth. And then you can look into this, and we can do comparative genomics and see what those differences are. And it turns out those differences are things like interacting with the host type 6 secretion system, motility, chemotaxis, attachment, nitrogen metabolism. So things that would be beneficial in the rhizosphere allow 2 of these particular strains to be more abundant than the other strains and to respond to the root. So it affects their abundance and it affects their ability to respond to the root. So it's not just the ability to use organic acids, it's ability to use the organic acids, amino acids, plus other things. So there's a hierarchy of traits that need to be considered. Okay. This is really to make the point that, again, most of the things that we're dealing with we've never seen and we've never grown. So we're relying on genomic approaches to predict the growth, the life history or the growth strategy or the niche of an organism. This is just some examples of papers here. And so the new approaches in high throughput assembly, binning, and annotation of microbial genomes are really pushing us into this field where most of our information now is coming from metagenome reconstructed genomes and not isolate genomes. Okay. This can be done at strain level, so we can now identify not just the OTUs, but the individual genomes and the loci in those genomes that are responding to a particular stimulus, like the root in this case. Okay. So Jill Banfield has developed a lot of infrastructure, and I'll show some data from that, working with Karthik Anantharaman. Ulash Karawas in our group is developing a workflow to go through assembly, binning, phenotype prediction, etc., and then interfacing with a new reactive transport model that Eric King is developing. He's sitting up in the back here. So how do we put all this together? This is just very quickly, we're looking at change in biomass and time. Going back to the automobile analogy, our engine, our biomass growth term, is it a fast grower or a slow grower? Our mu max we can predict from the genome. The bioenergetic driving force, the fuel grade, that's a function of the electron donor and electron acceptor that an organism can use. The Gibbs free energy, that's the power to fuel the cell. So that's the fuel grade. Then there's the kinetic driving force, how much fuel can the organism take in. So this is, Michaelis-Menten, this is your Km. And then a biomass decay term. This is, so this is effectively a dual monode with a thermodynamic constraint. So where do we get the parameters for this? Well, we can go dig the literature to find relevant organisms. That's always the place to start. We can do parameter estimation. We can take a community average and assume a distribution and sample from that. We can do, if we have a good distribution, we can do stochastic assignment of parameters. Or if we can isolate organisms, we cultivate and we do assays and we measure these things ourselves. Okay. The reason we are attracted to reactive transport models, and this is a nice quote from Carl Stiefel, it's where individual time and space-dependent processes are linked. The relative importance of individual sub-processes cannot be fully assessed without considering them in the context of other dynamic processes. So when we build a trait-based model for nitrogen cycling, those organisms are interacting with the carbon cycle, with phosphorus cycle, sulfur cycle, with metal cycles, etc. It gets very complicated pretty quickly. But to understand the true fitness of those organisms in the environment, you have to understand the environment that they're interacting with and feeding back to. Not just the biological environment, but also the chemical and physical environment. So reactive transport models are a great framework to build upon. So Carl had built CrunchFlow, which is mostly a geochemical reactive transport code with rudimentary representation of biology, and Eric has been working to improve the representation of biology in this, and one of the things is parameterizing certain things, reaction rates, building reaction networks using predictions from genomic data. So here is an example of a reaction network, and this is built based on a combination of geochemical information from the field, gene expression, information of gene expression, information from metagenomes, information of differences in abundance of organisms under different conditions. And that led to a hypothesis, which is represented as this reaction network. Each line connecting chemical species in this reaction network is colored according to an organism. In some cases, you see multiple colors. Multiple organisms are contributing to a reaction, and multiple organisms can contribute to multiple reactions. So that, in this case here, you have trait linkage, where an organism can be a denitrifier, but it can also be capable of cellulose hydrolysis. So its activity under denitrification impacts the rate of cellulose hydrolysis, so connecting the traits together. One thing we're working on at the moment, this representation here comes from a subset of genomes that we selected from a large set of 2,500 or 1,200 non-redundant genomes. But Eric is working on an approach now to systematically take all of the trait distributions and trait linkages from all of those genomes and build hypothetical organisms from that as opposed to representing just these genomes. I'll say a little bit about that if I have time. Another thing that Eric has implemented here is dynamic energy partitioning, and we heard about this. This is our trade-off. So energy available for catabolism can either go to growth or something else. First thing it has to do is go to maintenance and maintenance requirements for the cell. We're calculating that based on organism growth rate, based on published literature connections between growth rate and maintenance requirement. So that's the first thing that needs to be satisfied. Energy left over from that then can go into growth or into production of something else that takes away from growth, such as enzymes, nitrogenase, etc. Okay, and so what we want to do is put this network into an environmental context that's realistic. And this is in quotes because it's never really realistic, but somewhat realistic physical and chemical environment. This is a floodplain in Colorado. Water flows this direction, out from here into the river. Groundwater flows this way. There are some buried hotspots of organic material. The water table moves up and down, occasionally dips water into, or brings water up into these hotspots and brings oxygen back down. So there's a dynamic water table. And we're going to put this reaction network at different locations into this, into a slice of this aquifer and see how the community emerges and how biogeochemical processes interact. Okay, so this is just a kind of a simple representation. Groundwater is flowing this way, oxygen is diffusing this way, and we have some ammonia coming in from mineralization of organic matter up on top. We initialize this with an oxic region, a suboxic region, and an anoxic region, but things will look after itself after we get this going. And then we measured the fluid content coming in. So these are our boundary conditions. Each cell is initialized with the same biomass of each of those genomes that you saw. And then we put in some hotspots here, and you'll see how those emerge. So we're going to run this for 300 days, and you'll see different colors. Each of these colors then represents if it has a different color, it's a different microbial community composition. That's all it is. So we clustered all the microbial compositions, gave them a color, and just shows you how they evolve over space and time. Clearly, you can see the oxygen diffusion gradient of oxygen impacting the assembly of the microbial community. The hotspots are also impacting the assembly of the microbial community, and then the flow through those hotspots is influencing the assembly of the microbial community. Okay. So what does that mean? Well, we can look at reaction rates because we're calculating reaction rates at each time step and how they evolve over time. Here's a snapshot of reaction rates. You can see this blue area here. This is a nitrification zone. So what I didn't point out is that we initialized this with different types of nitrifiers, bacterial, archaeal nitrifiers that have different kinetic properties. But we see that the nitrite oxidizer and the ammonia oxidizer form a similar biomass in the same region. This is driven by thermodynamics, but also cross-feeding because it's necessary to cross-feed to have this reaction to occur. And the archaeal ammonia oxidizers are dominating because of the concentration of ammonia is favoring the and oxygen is favoring the archaea versus bacteria. So that's their traits are dictating their fitness. Further down here, this is something that we hadn't expected, but we have observed in the field. It was anaerobic ammonia oxidation, and that seems to be a decoupling because we have a decoupling of ammonia oxidation here. We have ammonia oxidizers, but the nitrite oxidizers are dropping down because of their lower affinity for oxygen. And that's resulting in a nitrite accumulation that allows anaerobic ammonia oxidation to occur. So we can see that occurring adjacent to area where the nitrite oxidation is happening. And we have evidence, transcriptomic evidence from the field that these are occurring. The hotspots here is where you see a lot of interactions and cross-feeding. Here we have cellulose hydrolysis occurring, followed by glucose fermentation, which is stimulating sulfate reduction. But we're also getting oxidation of sulfide and iron because we've got nitrate flowing in here, anaerobic oxidation. And again, we have genomic evidence, transcriptomic evidence for these being very important processes in the aquifer. Okay, so that's an example of what you can do with these types of models. And I think I already said this. So Eric is now working on taking, ideally what we want to do is not just build a model for a site from a metagenome from a site. We want to be able to identify generalizable features, generalizable properties of microbial genomes, what the trait distribution, the trait linkage in microbial genomes, and be able to create abstract organisms based on that honor those evolutionary processes that have resulted in those genome, in those those patterns of trait linkage. So acquiring metagenomic from a site or compile all data, analyze the genomes for traits and trait linkage. So in this case here, each column is represents either a pathway or a set of genes coding for those reactions that we represented in the previous network. So you can see that it's not uniform. This patchiness here. We form clusters. Each of these clusters would form what you might call a guild. That contain a guild based on trait linkage. Okay, and so based on this and chemical reaction libraries, you build a trait repository connecting pathways to chemical reactions. Here, then, what you're trying to do is reduce the complexity of this to retain a proportion of the information. So you're going to reduce 1,200 genomes or so down into a couple of hundred at this point here, right? But still representing most of the variance in trait linkage distribution, right? That's the goal. And then moving into building representative organisms based on how satisfied you are that these simplified clusters represent the original, represent the information in the original clusters. Okay, I'm not going to show anything on this, but this is something that I think is really important to work away, to work through this systematic approach so that we can take advantage of genomic data from other data sets and not just from our own systems. Okay. Okay, that's pretty much it. I just want to go back to this representing the complicated and the complex. And really we need to be able to validate these omic proxies for traits, determine the trait distributions and the trait linkage. That requires omic, but also cultivation. I really think it's important to use these thermodynamically based performance filters. The differences in free energies available between reactions is huge, and that can explain a lot of the variance in microbial community dynamics. And we need formal mechanisms to make the microbial diversity tractable. So this formal mechanisms of condensing those clusters into more tractable units that we can represent in a mathematically feasible way in models. And then this last part here, this is a DOE like to use this word MODEX, but it's just model experiment integration. And so that you begin with your initial model, simulate, observe, manipulate your system, see when your model failed, improve, and go back. Just iterating through this process and using these model outcomes to identify experimental parameters that are that are the real test of your model accuracy. Okay. Okay, and with that, there's a lot of people involved in this sort of general project. Kate Zalnina did most of the metabolomics work. Eric has been developing BioCrunch in Ulash, and Hee Jung did most of the bioinformatics. And Eric, Jin Yun, and Yiwei are here actually at this as well, so it'd be great people to talk to and ask other questions too. Great, thanks.